Dear students, in this module, I'm going to introduce you to the topics of similarity and identity. Why do we need to study these topics? If you may remember, we're trying to compare two biological sequences. By biological sequences, I mean either a set of DNA sequences or RNA sequences or protein sequences. So when you're trying to compare two sequences from any one of these types, then you would want to evaluate. You will want to check how good is the comparison between the sequences. Towards that, we need to know what is similarity and what is identity. To begin with, I'll talk about identity or sequence identity as it is called. Sequence identity is the number of nucleotides in case of DNA or RNA or amino acids in case of proteins which may match exactly between two sequences. If we match 10 nucleotides from a DNA or an RNA sample with another 10 nucleotides in another DNA or RNA sample, then we can count how many nucleotides are there in each one. We can also look at which types of nucleotides are there in each one of these molecules, and we can also look at their arrangement. So let's take a look at an example. Here you have two nucleotide sequences CATGCTT. So I have numbered this to be sequence number 1 here, and CATGC, that is sequence number 2. So I would like to compare these two sequences and evaluate their similarity. So how can I proceed? The first thing that I need to do is I need to count how many matches exist between these two sequences. So as you can obviously count, the first nucleotide C matches, the second nucleotide A also matches. Similarly, the next three nucleotides T, G and C, they also match. So in total, you have 5 nucleotides that are matching exactly between these two sequences. Next, I'll have to calculate which sequence has a smaller length. You can simply count the number of nucleotides in sequence number 1 and sequence number 2. In sequence number 1, you have 7 nucleotides here, and in the second sequence, you have 5 nucleotides. So you are supposed to pick up the smaller length. So obviously, you will select 5. Next, you need to apply this simple formula wherein identity equals to the number of matches, that is 5, divided by the smaller length. As you just saw, the smaller length was 5 because sequence number 1 had a length of 7. So you selected 5. So now you have 5 over 5, and you have to convert it into a percent. So therefore, 5 over 5 is 1, and 1 into 100% is 100%. So if you want to talk about the identity of these two sequences, they are 100% identical. So the points to remember in this case. Number 1, the gaps are not counted. So if you have a gap in the sequence, then that is not considered as a match, and that is left out. Second, identity measurement is always made on the shorter sequence. As we just saw in the previous example, the shorter sequence was 5 nucleotides long. So we need to consider 5 as the smaller length. These 2 points are very important while computing the identity between sequences. Next, similarity. So what is similarity? So similarity essentially is the result of matching and transforming one sequence to another by finding the smallest number of edit operations. So let's say if you had sequence 1 and sequence 2, then you would want to compare these two sequences by sliding them against each other such that the number of modifications you have to make in one of these sequences in order for the rest of the nucleotides to match, you have to do minimum number of deletions, insertions, and substitutions. So this is what is called edit operations. So we will look at insertions, deletions, and substitutions later, but what you need to remember is that you need to cater for them. So therefore, to compute sequence similarity, you need to first align the two sequences in order for you to obtain the gaps and mismatches. This can of course be done by using pairwise sequence alignments. So in conclusion, identity is the count of nucleotide or amino acid matches that are exactly matching between the two sequences, and the gaps are excluded from this exercise. Similarly, similarity is a different measure in which you have to compare the two sequences after aligning them with each other.